Thanks. Okay. Welcome back, everybody, to our fourth session today for this year's IGRM. We've had some great talks so far, and we've 4 more to come. I'm Betty Higgs, retired from University College Cork, but still seem to be working there too. And my co-presenter, Luke O'Reilly, who is also in UCC. So our first talk for this session is from Devon Smith, and Devon is joining us from Ohio in the USA, so in a different time zone. And the talk is about nitrate sources in rivers Shannon and Corrib.
Thank you, Betty. I'll just share my screen here. Okay. Can everyone see?
Yes.
Screen. Great, thank you. Well, again, thank you for that introduction. As Betty said, I'm from Columbus, Ohio. I'm a PhD student at The Ohio State University, and today I'll be presenting a section of my dissertation work that focuses on identifying sources of nitrate in rivers Shannon and Corrib and select tributaries. This is part of a larger work that our lab at Ohio State has been conducting with colleagues at NUIG for the past 3 years to look at the geochemistry of Irish rivers. And so I'll be starting the talk today with discussing motivation for this work and the sampling campaigns that we conducted in 2019 and 2020. I'll also talk about the utility of stable isotopes of nitrogen and oxygen to discern the sources of nitrate and present the data that we collected and also show fluxes of nitrate within these river systems. And then I'll be ending with implications and future opportunities for this work. So the first question is, why study nitrate in Irish surface waters? And one of the big reasons is the EU Water Framework Directive. Many of you probably know that this is a program implemented to protect water resources across Europe. And it's important to protect these resources for both ecosystem and human health. Nitrate is an essential nutrient in surface and fresh and marine environments, but it can also cause eutrophication when there's too much. And this is exacerbated in marine environments because nitrogen is the limiting nutrient. And so it's also important to look at for human health because surface water is a main source of drinking water in Ireland, and elevated concentrations of nitrate can cause water degradation. And so because of these reasons, the EPA has been monitoring concentrations in Irish freshwater resources for several decades, but monitoring concentrations is only half of the equation. The other half is looking at the source of nitrate so that successful mitigation techniques can be implemented. And so in order to look at the source of nitrate being discharged to the west coast of Ireland, we sampled the River Shannon in 8 locations, 5 Shannon tributaries, and 5 rivers within the Corrib watershed. During our sampling campaign in October of 2019, we collected samples to analyze for stable isotopes of nitrate. And during our other sampling campaigns, we collected surface water samples to analyze nitrate concentrations. You can see on the map here that the sampling locations for each spot where we sampled for nitrate stable isotopes are plotted in white, and the primary land uses in Ireland, which are pastureland and peatland, are shown in yellow and green. And these land use data are from Leiden and Smith 2014. And knowing these land uses and recognizing how expansive they are across the country will be important for my discussion of stable isotopes today. But before I jump into presenting my data, I want to talk about how isotopes of nitrogen and oxygen can be used to discern the source of nitrate within an aquatic system. And we look at these stable isotopes in the delta notation, which is the ratio of the heavier to the lighter isotope of the sample minus the standard divided by the standard and multiplied by 1,000. And this gives us a per mil notation. And when talking about stable isotopes of nitrate and nitrate, so oxygen and nitrogen, it's useful to refer to them in terms of heavy and light. Heavier samples trend towards a more positive value, and lighter samples trend to a lower or more negative value. And so if you plot the isotopic composition of your nitrate sample on this plot, you can see that there are ranges that indicate the initial source of that nitrate. And this plot was first generated by Kendall et al. In 1998 and is very, as I said, very useful for indicating nitrate source. But there are certain sources that overlap, and so sometimes it's necessary to use other spatial analyses and/or conservative geochemical analytes to discern the source of nitrate within an aquatic system. So if I plot these, the isotopic composition of the samples we collected on this figure, we can see that all the samples fall within this nitrification box. And so the oxygen 18 values for those samples range between -1 and 6 per mil, right here. And then the nitrogen 15 isotopic composition ranges between 5 and 11 per mil. So since we can see that all the samples fall within this nitrification box, I'm not going to talk too much about the oxygen 18 values of the samples today. Instead, I'm going to be focusing on the nitrogen 15 values to discern if the samples reflect a soil ammonium signature or a manure and sewage signature. And if I zoom in to the nitrification box, we can see the samples a little bit better. And now what I've done is I've color coded the samples based off of the pastureland area in the drainage catchment. So for each sample location, I delineated the drainage area and then calculated the percent of pastureland area within that drainage basin. And I've color coded them based off of that pastureland area. And so we can see that there's a relationship between a heavier nitrogen 15 signature and the percent pastureland area in the catchment, which would indicate that there's a correlation between pastureland area and a manure and sewage source for nitrate in the Shannon and Corrib systems. I also did the same with peatland, and this shows a little bit of a different trend. Now the samples are color coded by the percent peatland in the drainage basin from 0 to 60%. And the lighter isotopic composition of nitrogen 15 reflects a greater percent peatland area or a soil and ammonium signature. And so again, while this is very useful for looking at the stable isotopic composition of nitrate in a system and for relating it to source, you still need a statistical analysis and to look at it spatially to fully understand mixing and processes within the system. And so what I did was I calculated R values or correlation of coefficients to look at the statistical relationship between nitrogen 15 and land use and nitrate concentration and land use. And so what the relationship between these values show is a positive, they show values of 1 being +1 to -1, where a positive 1-to-1, +1 is a 1-to-1 correlation between the two variables, and -1 would be a perfect negative correlation between the two variables. So the nitrogen 15 and peatland have a negative relationship, which coincides with what we saw on the previous slide, where a lighter or lower value of nitrogen 15 corresponds to a greater percent peatland in the drainage area. And the positive relationship between nitrogen 15 and pastureland represents also what we saw in the isospace plot, where a heavier signature of nitrogen 15 corresponds to a greater percent of pastureland in the drainage catchment. There's also a negative relationship between the nitrate concentration of and peatland area, and that's because peatlands have a higher retention rate for nitrate. And there's a positive relationship between pastureland area and nitrate concentration, likely due to overland and flow and subsurface discharge into river systems, leaching nitrate from pasturelands. So if we look at the sample spatially now on this map, this is the same map I saw a few slides ago, but now it's zoomed in, and the sampling points are color-coded based off of the nitrogen-15 signature. So a lighter color corresponds to a lighter signature or an ammonium-15 value, and a heavier signature or a darker color corresponds to a heavier signature or a manure source. So starting at the top of the Shannon, we can see that all of these values have a light isotopic signature, and these also drain a higher percent peatland and lower percent pastureland. But if we travel down the main stem of the Shannon, the isotopic composition grows heavier and reflects more so a manure source, particularly coming from the tributaries draining from east to west into the main stem that have the heaviest sources that we saw in the system. This results in a heavy source being fluxed out into the Shannon estuary. In the Corrib watershed, the rivers Robe and Clare also reflect that heavier nitrogen-15 signature and reflect also a manure source. And that is what we see also going out into Galway Bay from the Corrib River. So now we've seen the statistical relationships and the spatial patterns of nitrate sources within these watersheds. But as I said earlier, looking at the source of nitrate is only half of the equation. The other half is understanding the amount of nitrate that passes through that system. And so what I did was I calculated fluxes of both of nitrate at certain sample locations along the Shannon and the Corrib watersheds. Or rivers, I apologize. So what I have here is I've calculated nitrate in kilograms per day of nitrogen as nitrate and during sampling campaigns of October 2019 and March 2020. Along the x-axis is the nitrate flux values, or along the y-axis the nitrate flux values, and along the x-axis are the river sample locations, and they're spaced in a transect from north to south of relative distance where we sampled. These flux values were calculated using the concentrations measured during our sampling campaigns and using discharge data from the Office of Public Works. So if we can recognize a few patterns, both through October and March of 2020, the flux increases downstream. So the further downstream you travel, and more water that volume is added to a system, which results in an increase in discharge and therefore an increase in flux of nitrate. We can also see that in March of 2020, all of the flux values during the sampling period were greater than their corresponding points in October of 2019. And many of you may remember last February and March were very wet in Ireland, resulting in more water being added to the system and therefore an increase in discharge. So in addition to a greater nitrate concentration seen during the sampling period of March of 2020, the amount of discharge is greater. And just to give you a numerical relationship for that, the average discharge at Banagher during our October 2019 sampling was about 320 cubic meters per second. And then that's at that same location, it was 590 cubic meters per second in March. So as I said, a lot more water traveling through the system and therefore increasing the flux. We can also see a similar pattern in the Corrib watershed. Once again, I calculated flux in terms of nitrate as nitrogen in kilograms per day. But now, in addition to the October 2019 and March 2020 sampling periods, I have an additional one in March of 2019. And along the x-axis are the river locations plotted in a north to south transect. And so all of these rivers, again, were the flux for these rivers were calculated based off of samples taken and concentration values analyzed, and then the discharge values from the Office of Public Works. But we weren't necessarily able to collect samples for stable isotopes of nitrate at all of these locations. For example, the Owen Riff, which has the lowest flux at each location, drains, 68% peatland area, but we were not able to sample nitrate isotopic composition. However, that low flux corresponds to that R value that I showed a few slides ago that shows a negative relationship between nitrate concentrations and peatland area in the drainage basin. The rivers Robe and Clare, which are those two rivers that drain the east side of the Corrib catchment, had the greatest variation in flux throughout the 3 sampling campaigns. And the Corrib River consistently had a high flux because this is at the mouth of the Corrib watershed and has greatest volume of water traveling through it. With that said, there is some estimation going into the discharge values for the Corrib discharge, so there could be some considerable variation in these flux values that were calculated for that location. But overall, I can draw a few conclusions based off of both these flux values and source values for the Shannon and Corrib watersheds. And the first one is that the flux increases downstream, and we saw that in both watersheds due to the increase in water volume and therefore increase of discharge in the system. And it also increases in flow due to different precipitation changes. Nitrate is tied to the sources of land cover in each of these systems. And for the Shannon watershed, that means that it's primarily a manure source. And we saw that particularly mixing in the main stem of the Shannon as the tributaries coming in from the east had a heavy nitrogen-15 signature. In the Corrib watershed, we also saw that signature of rivers draining pasture lands represented in the flux out to Galway Bay, as the rivers Robe and Clare had the greatest flux of nitrate and also the heaviest signatures. And so overall, this has important implications for implementing mitigation strategies to reduce nitrate concentrations in the Shannon and Corrib watershed and delivery to the west coast of Ireland. This source and flux analysis allows policy to be focused on locations that have a large amount of nitrate traveling through them and use methods to reduce this just diffuse pollution. And so future opportunities will involve investigating these nitrate isotopic compositions and sources in relation to groundwater input into these systems, and also using other geochemical analytes to analyze the relationship between nitrate and land use as well. And so I'd like to thank all of the individuals and organizations who help make this work possible, both for financial, academic, and logistical support. Thank you.
Thank you, Devin. Thank you, Devin. That's great to see such, that quantitative work going on an important topic. Luke, do we have any questions?
Yeah, thanks for that, David. Yeah, we've got one there coming in YouTube from Patrick. Patrick asks, how will cessation of commercial peat harvesting and reflooding of bogs in Shannon catchment impact on nitrate fluxes? And that's been a very recent case here in Ireland.
Yeah, so obviously changing the water table can have great implications for affecting nitrate fluxes. So it's primarily dependent on the oxidation conditions, right? So nitrification occurs during oxidized environments, and so when you have a pulse of water in a system following a period of oxidation, you're going to get nitrate release from that system, similar to what we see in the pasture lands, where you have drainage from overland flow or just subsurface flow into a river system. And so I think that that's going to be particularly important following dry periods or, refilling these peatlands that were previously mined. Yeah.
Thank you. Question from Kuhn. Great talk. Did you get a chance to look at long-term trends of nitrate values from EPA studies in the same areas? Also, any recommendations on mitigation or sign of improvements?
Yes, so that is something that I will hopefully, I would plan to be doing in the future, looking at EPA nitrate values to look at this a long term. And in terms of, and I'm sorry, the second question was mitigation suggestions or?
Any recommendations on mitigation or sign of improvements?
Okay, so I can't speak to sign of improvements, but I can speak to suggestions for mitigation, and creating buffer zones is very important to reduce the flux of nitrate from pasturelands into river systems, and particularly to reduce overland flow. When you're creating a buffer, it'll prevent that nitrate leaching directly into rivers.
Thank you very much. I think we're on track there. I might hand it back to you there, Betty.
Thanks, and thank you very much, Devon. Our next talk, we're moving offshore. So we have integrated geophysical investigations of deeper stratigraphy of the Irish Rockhole Basin from north to south. And we have to present this, Gaurav Toma and his team.
Thank you very much. I'm just going to share my screen. I hope everybody sees it.
Yes.
Thank you so much, Betty. I'm going to present about the integrated studies, as you have read just, in the north and the south of Rockhole. So we have included seismic, magnetics, and gravity to study the crustal to upper mantle structure in the Rockhole Trough. This is the outline of my talk today. So today I will show you the topography and geological map of the area. Then I will talk about a little bit of the methodologies. And then I will talk about some crustal to upper mantle structure of the northern Rockhole Trough, where I have applied travel time tomography on 2 seismic profiles and gravity modeling on 4 seismic profiles to see the variation of the crust, the depth of the moho in the Rockhole Trough in the north part. And then I will show you some results about some travel time tomography and gravity and magnetic modeling results of para-volcanic system in the south part of the Rockhole Trough. And then I will conclude my presentation. So here these are the topography and geological map of the Rockhole Trough. So first question is like why Rockhole Trough? So I could only explain that the Rockhole Trough is a very natural laboratory to study the rift system and mental serpentinization and the formation of the magmatic and non-magmatic basins. As we can see, here these are the seismic profiles I will be showing some results from. These are the 4 seismic profiles in the north, and then I will show some results on this profile where my cursor is at the moment. So here this profile, why this profile is because it is crossing the paravolcanic system. Here there are 3 paravolcanic system is previously observed. So this line serves to see the structure of these things well in the south part, and then the north part of the Rockhole Trough is flooded by the silts. So then we will explore a little bit about using these 4 seismic profiles across the basin, where I applied travel time tomography in the beginning, and then I have applied gravity modeling on the 2 others, including the G2 in the north part. So first, I will talk about the methodology. So this is basically the one shot I'm showing you here. So then what we do is like we do the downward continuation. The downward continuation is the process where we put our sources and the receivers at a certain level above the seafloor, because here the water depth in Rockhole is almost 2 kilometers, 2 to 3 kilometers. So that enhances our signal of the refraction. As you can see, after putting our source and the receiver at the seafloor, here we can see the first arrival we can pick till 4 kilometers, but after the downward continuation we can pick our first arrival around like 1 kilometers. So it enhances the refraction and we can see the shallower information very well. And then we apply travel time tomography to get the velocity of the structures where after picking these first arrivals on downward continuum data set. So then after getting the velocity for gravity modeling, we need to have the density, so we use this Neft-Rake relationship and this relationship for the sediments, and Neft-Rake curve for the crustal velocities. So this second relationship we use for the sediments because it is specifically computed for the sediments by using thousands of well logs. So now after we have the density and we could also compute the velocity, now we will see how we do that. So we in the north part, I use these four seismic profiles where they are very well with where we have the lots of silts here. So this is the line I'm showing here. This is the seismic section of profile PAD16, the second number one. So here when we see the silts are getting kind of the flat, we can see some basement reflections as well as some sedimentary reflections beneath the silts. So here what we see, so here if I will also want to go back, so where you can see our refraction after like 9 kilometers in this area, we have no signal in our reflection because it indicates that we have the sediments beneath the structure that is recorded here. So when we have silts, so we rarely get the signal of the structure beneath the silts. So now we apply the travel time tomography. This is the final velocity model after 14th iteration. And here we can see that our velocity model, we only can have the resolution by, till 6 kilometers. And we can see that the velocity model also follows the sill structure as well in the velocity. And we get the velocity from 1.8 kilometer per second to 4.5 kilometer per second of the sediments in the local area. That match very well with the previous studies. So now I move to the initial and final residuals. So here, when we take in the travel time tomography, this is the initial residuals where we start with our velocity model, initial velocity model, and then we converge to this final residuals. Here we can see, so our final mean residuals reach to the 18 millisecond, where in the travel time tomography we include some uncertainties of receiver source position, bathymetry, and travel time. So that is include like almost 16 millisecond, and our chi-square is to 1.4. That means our mean residual time is 18 milliseconds. So we get a very good match with the picks we have. I have picked manually for like for this is like 5,000 shots. So we pick every fifth shot. So it's like 1,000 picks for each line manually we have done. So now, so our velocity is very good to explain the data set. Our residual also shows that. Now I move to the second line. This is the northernmost line in my study area. So here we can see some basement reflections as well as some force-folded structures where the sills are kind of using some force to force-folded the sediments. So there goes like uplifted of the sediments. Here, and then after applying the travel time tomography, we get the velocity model that is these are like also down to 6 kilometers. So now, because by the seismic we can only go down to 6 kilometers. So now we include gravity modeling to understand the deformation of the trough. That can be obtained by gravity modeling. So here I show you the gravity modeling of line 16. So where I have shown the velocity. So then these are the time section picked all the horizons from the seismic, and then these are the depth converted horizons with including the velocity converted to the density, and with the density we used here. And this is my final and the observed and the final gravity anomaly using the model we have here. So now we also can measure the Moho depth in the local trough for this line. So that also give a chance us to compute the stretching factor. So stretching factor we compute by using the initial crustal thickness that is almost 30 kilometers in the local trough, and then we use the basement reflection to compute the current crustal thickness. So here for this profile we observed a stretching factor that goes maximum stretching factor goes more than 6.5. Now I will show you the results of gravity modeling of line 18. So here the same as like the travel time section till 12 seconds, and here I show the depth section till 22 kilometers depth. And here by using these horizons picked by the seismic, using the velocity we obtained and the crustal and the mental velocities we used by using previous informations done in local draft. So here also we get a chance to measure the stretching factor that goes also here more than 6.5. Now I move to line number 14. Here you can see the location of the line. Here also we match the gravity very well with the observed one by using the picked horizon from the seismic and then convert it to a depth horizons, and using the density of the converted velocity. So now we can see that here also we have the stretching factor maximum max stretching factor that goes more than 7. So why we are computing the stretching factor that we will see later in the concluding remarks, why they are important. So now I move to line 12. That is very important because the Moho structure varies here quite well. So we get like shallower and where like that is following the gravity as well very nicely. So this is a very interesting line. So now these are the results in north part of the Rocco, and here as well as we also compute the stretching factor that goes more than 7. So now I move to the nature of paravolcanic system that is here. So previously we have imaged like previously authors have imaged 3 volcanic systems. Here these are the shown by the gray line like this. So to image that we use PAD line 28, but the part of one that closes this one, but only half of the line that include almost 10,000 shots, because this line is 300 kilometers long. So then these are the seismic profiles. So here we see one in the beginning, that is this guy that is not previously explored, and these are all other like 3 volcanic system, all the volcanoes that previously imaged. So these are the 350 kilometer long profile, and it contains like 9,000 shots. The receiver spacing is 12.5 meters, and shot spacing is 37.5 meters. So for doing travel time tomography, we divided this profile in 2 pieces, like 1 to 160 kilometer and 162 to the end. Here, this profile include only half of the profile what I had shown previously. So, and this is the final velocity model obtained by using the travel time tomography after 14th iteration. So here we can see in this part there is almost no sills. So that's why we can see this basement reflection very well, as well as the structure that goes deeper down to the 7 second. So this is the benefits of not having the sill, so that we can have the deeper image. But this data set also failed to image the Moho because I don't know why. And so now, as you can see, we image this structure, this volcanic center very well, as like we can also image the flanks of the structure, and the velocity goes to 4.7 to 4.8 kilometer per second. That match very well with the previous studies in this area, as well as in the Porcupine and in other areas in this region. So now this is the initial and final residuals. So these are the initial residuals, and then our final residuals goes like down to very that is matched very well with the mean residuals time with the data uncertainty, where our chi-square reaches final chi-square reaches to 1. That is the goal of the travel time tomography. So now I move to the other part of the profile. So here we see there are like the other volcanic centers are out of our reach, but this one we have imaged well, and the shape of this volcanic center as well. So here we had seen that there in the southern part there are a few 3-4 volcanic centers. So to prove that, we need a magnetic modeling to support that if we are saying is like it's really magmatic or is non-magmatic, and we can see after matching the gravity. So we also put like these anomalies separately in the GeoSoft software where we match our magnetic data with observed magnetic data with a synthetic one, and here the magnetic anomalies goes to around 2 ampere per meter. So here this magnetic modeling also confirms that these structures what we have seen in the seismic images, they are volcanic. So then it comes out the question like why there are so much magmatism in the southern part of the Rockel trough. So this, and also like here in southern part as well, we also compute the amount of extension that goes more than 6.5.
It's 2 minutes.
Now we conclude that we have performed like downward continuation, and then we have done some pre-processing of the data set before applying downward continuation. Then we have computed the sedimentary structures down to 6 to 7 kilometers, where the velocity goes to 1.8 to 4.5 kilometers. And in the absence of long offset data, we also included the gravity modeling as well as magnetic modeling to distinguish between the magmatic and non-magmatic structures, and gravity modeling to get the Moho structure in the absence of long offset data set. So we have obtained upper to lower crustal, lower crustal to upper mantle structures. So here now I was talking about the stretching factor. This maps contains a very good information that is obtained by the modeling, synthetic modeling. So this black curve shows that when our stretching factor reaches to 3.5 to almost like 4 to 3.6, so our mantle serpentinization begins. So, and we have obtained for the Rockel trough is more than 6 from north to south. That means we conclude that it favors the mantle serpentinization hypothesis for the Rockel trough, as well as we have obtained some relation, as well as these graphs also contain some information by using the melt thickness and the rift duration. But in the Rockel trough, if we have obtained 3 to 4 kilometers of the thickness of the melt, so that will be that may need very fast rifting, as similar as like Porcupine, or it may be possible that the southern Rockel trough was at the verge of creating the oceanic crust before breakup. Now there are some perspectives of the study. So as we can present a 3D geological model, we have several seismic profiles so that we can combine them and can produce a 3D crustal to upper mantle model for the Rockel trough. And it will be very good to see the relationship between the Porcupine and the Rockel trough, because we have observed magmatic activities in the southern part of the basins in the Rockel trough and as well as in the Porcupine. And for the better understanding, we should be doing pre-stack depth migration, and then we will need, as like we had seen, the 10-kilometer long streamer failed to serve the purpose to image the deeper part of the basin. So that's why we need some long streamer, long wavelength data set for fully understand the basin structure. I will acknowledge for our founders, IGRM, SFI, PIP and IS, and we will also thanks to PAD for providing us the data set, as well as Satish for helping us in tomography and downward continuation. And this is the third, and I am open to question. Thank you very much.
Thank you, Corrib. Very interesting there. I think we have time maybe for one question, if there is one, Luke.
It looks like that the chat at the moment is blank. I think Gaurav just covered everything there, but I know this is kind of a topic which is of interest to you, Betty, so maybe you might have one.
Well, I was very interested to see that conclusion that perhaps we were at the edge of the forming the oceanic crust. Can you say something, just describe the topography of the moho there, Gaurav? That looks very interesting.
Yeah, it is. It is very interesting to see that we have observed very good deformation in the moho, because it varies from north part to the south part. So that means it is very interesting to make a 3D model and to see and to look at the deformation of the whole basin.
Yes. I know that you're building on the work, great work by the others in your team. So seeing the integrated techniques is really paying off. So thank you, Gaurav. So we'll move on now to our third talk. So now we have carbonatites as chroniclers and critical resources, an example of the Gifford Creek carbonatite complex, and we're moving to Western Australia for this talk by Paul Slezak and team.
Paul, you might be on mute, maybe. Hello,
Paul?
Oh yeah, there we go. Sorry. Cool. Well, thank you very much for the introduction. That pretty well covers it. For something a little different, we'll move to the opposite side of the planet and talk about some work I did during my PhD with my advisor Carl and my colleague Timur. And really what we're going to do today is we're going to take a look at carbonatites and kind of why they're useful and some of the things you can do with them. So to start off, they're just uncommon rocks. They typically have over half of them are composed of primary carbonate minerals, and they typically occur with a lot of other kind of weird rocks and alteration assemblages, which we'll get to see here in a moment. And with that weird geochemistry, we also get a lot of kind of weird and intimidating rare earth-bearing minerals, some cool ones that we saw earlier. And these same particularly rare earth-bearing minerals, they're what make carbonatites important sources for critical metals, but they also make them really useful for geochronology and geochemistry. And for those of you hoping to look at some of these admittedly intimidating minerals, we won't really be looking at that. We'll actually be looking at a few more common minerals, zircon, monazite, and fluorapatite, although they will look probably a bit different than many of us are used to. So just real quick, there's definitely more than 500 carbonatites worldwide, and there's about 100 rare earth occurrences, deposits, mines, stuff like that. And this just kind of highlights some of the major deposits and mines in the world today. And we're actually going to move down to the Yangibana light rare earth deposit here in Western Australia. And that's hosted in the Gascoyne province, which is a Proterozoic age suture between the Pilbara Craton to the north and the Yilgarn Craton to the south. And the big takeaway here is that the Gascoyne province is kind of divided up into these big shear zones and fault zones. And these, through seismic surveys, these big faults have been found to go all the way down to the mantle and offset the moho in a lot of instances. In particular, this big fault here is the Lions River Fault. So just moving into that study area, that little lobe effectively hosts the Gifford Creek carbonatite complex, which is a series of paralkaline dikes and sills hosted in this kind of 400 square kilometer area between the Lions River Fault and the Baldwin Liniment. And within that complex, there is a series of discrete deposits that collectively make up the Yangibana light rare earth deposit. And, well, I guess really moving on, as you saw from the map and from this figure, we'll jump back and forth. There's a lot of different rock types going on there. There's these calcite and dolomite, carbonatite dikes and sills here, known as the Lion River sills, and they typically occur in the southwest here near the Lions River Fault. There's magnetite biotite dikes. Some can argue that they're actually phosphorites. and they're really located here in more the southeast region. Then we also get these kind of weird silica-rich paralkaline veins, which are orthogonal to the Lyons River sills. And then really what was discovered in drill core fairly recently are these anchorite siderite carbonatites, or ferrocarbonatites. And they're located at depth beneath their altered versions called ironstones, and they're really found around the periphery of the complex there. And then associated with all these rock types, we get some interesting alteration assemblages like phenites, which we can see here is kind of this K-spar-rich magnetite-biotite alteration halo around the ferrocarbonatite. And then we also get glimmerites, which are just biotite, magnetite sort of stuff. And then again, the ferrocarbonatites, which altered ironstones. But really what we're going to take a look at here is kind of more of these primary units, and in particular, their rare earth phosphate mineralogy. So we can see from these pictures here, and we'll go over where these are all from, monazite's quite large. You could see it in hand sample in a lot of cases, but it's large, big euhedral bladed crystals, and it's often intergrown, like you can see in these two pictures over here, it's intergrown with apatite, which is again, large and very abundant. So in a normal instance, if we want to date like a granite or a basalt or something, we have to do a lot of mineral separation, but the luxury of the carbonatite system here is that we can do a lot of work in situ. So a lot of the work we did was just laser ablation stuff for uranium lead, and then in situ laser ablation multi-collector analysis using a Neptune. So all the data that I'm going to talk about here, that's really where it's coming from. So again, these samples are Yangibana North, Yangibana West, and Fraser's, and they're just located up here, Yangibana West, which is actually a bit north of Yangibana North. And then, so these two over here in the far west, and then Fraser's here down in the east-southeast part of the complex. So right before I started my PhD, there was a little bit of work done, and actually down in the south here with the Lyons River sills, which has been the big focus of everything, because they're well known. And there was some age dating that came out with monazite and apatite about 1050, 1075 million years. And they're like, oh, it's associated with this large igneous province way off to the east, case sorted. Well, through drilling and other exploration, they found these new rock types. And so I got a PhD out of it. And one of the things, again, with this luxury of being able to do in situ work that I was able to do was a lot of samarium neodymium work. So I was looking at the petrogenesis of everything. So we did neodymium work. And with all the monazite and apatite, we were able to do a lot of isochron work. So most of our samarium neodymium isochrons came up 1250 or 1260 million years, except for this guy here, which was the only one, and it came out much younger, closer to those original uranium lead ages. But given the amount of samples, it's not really an isochron, it's more of an errorchron. So it's not the most trustworthy thing. So we did that, but we're kind of like, the stuff that they dated, that's from different rocks than the stuff from previous work is from the Lyons River sills. We're dating stuff a bit further up north. Let's keep going with things. So again, we've got this big monazite. We can do a lot with it. We just did uranium lead dating. And we did this sample and things were great. Everything's within uncertainty of the samarium neodymium dates. So we kept going with our other samples and things weren't so good. The uranium lead ages were all outside of uncertainty, not only with each other, but with the samarium neodymium data as well. So as you can see, these three ages here are pretty widespread, and the bunch of other samples that I did too was pretty much the same result. So that's a bit of a problem because now we can't really trust monazite and some resetting and stuff is going on. But then we found some zircon, which is which is even more robust than monazite or apatite in a lot of scenarios. And you can see these zircons here, they're not really what you'd expect out of a granite. You've got this nice, one, they're very large, and two, you've got this distinct dipyramidal shape here. And they've also got some interesting chemistry as well. And they were found in these kind of alteration halos, these little black lines and zones down here, which are a glimmerite alteration assemblage associated with this kind of beige and buff colored anchorite siderite carbonatite. So we know that they're associated with the carbonatites, and we were able to find those pretty much again all over the deposit. So doing uranium lead on those samples, we got a completely different age of 1360, 1370 million years, but everything was concordant, and it didn't matter where the zircons came from. They were all very, very consistently this age and all within uncertainty with each other. So this was also interesting because there wasn't any recorded tectonic events going on at this time, so we were kind of like, uh-oh. And so when we plotted up all the data, the zircon data is here in pink, and before you're like, oh, we've got this really early age here, that's from the granites, not which these others, the Gifford Creek carbonatite complex intrudes. So this all makes sense, but really this kind of strip right here of alkaline magmatism hadn't really been identified. And that's because everything was done on monazite and apatite, and really you didn't see this big spread of the data, which isn't worthless. It's actually quite useful because, as we can see here, the uranium lead from monazite is actually recording a lot of different tectonic events and orogenies and stuff like that. The youngest of which is kind of like the onset of the breakup of rhodinia. And then there's some interesting stuff going on that a lot of this samarium neodymium data is all pretty much within this big tectonic event, which is called the Mother Buchan tectonic event. And then we get this interesting kind of dissociation between samarium neodymium and uranium lead, which in itself is interesting. But again, going back to this kind of 1370-ish age, not a whole lot's going on in Australia at the time. At least not from an orogenic standpoint, and certainly not inboard of anything. But if you jump back a bit to a bigger tectonic setting, at about 1.3, 1.4 billion years, the West Australia craton and the North Australia craton, and however you really assemble these two plates, they're separating from the North China craton. And coincident with that is also the formation of Bayan Obo, which is the largest rare earth deposit in the world. So that's admittedly a bit more proximal to the separation of these two continents, but it's interesting to note that even in a slightly more intercratonic setting, you do get reactivation and kind of some decompression melting to form carbonatites that are of note as well. But that's kind of a different talk. But it is important that there is this big event here happening. So now that we know when everything formed, we did some lutetium hafnium, again in situ on the zircons, but because we're mostly operating with neodymium values, we had to go to the literature and kind of convert that using the mantle array to neodymium values, and then that corresponded well with our bulk rock neodymium values. So everything's the same, and it's from the same event. And then taking that data, what we did was we plotted up our epsilon neodymium values, and this is pretty much when everything started. And this dashed yellow line is how it would evolve through time. And then, in addition to that, what we did was we took the neodymium values for everything at its younger resetting ages and then plotted them. And they weren't different. They fall along this line. And this is actually really important because when we model kind of an idealized enriched mantle situation, the line should go like this. And the reason that everything falls along this trend is important is because if it were to kind of come back up, or at least be dispersed towards the enriched mantle line, we would infer that maybe all those different rock types that we just saw were from different events. Maybe you had a refertilization of the mantle and more magmatism and more eruption and stuff like that. But in fact, it's just one discrete event, and it's just kind of evolving through time. So again, this just kind of reiterates that trend. Things had been reworked, probably in a closed rare earth system. That's why everything kind of stays along that line. The discrepancy in ages is really interesting because it records these big tectonic events. And the decoupling of monazite, at least for uranium, lead, and samarium neodymium, is interesting, if not cautionary, in that looking at the chemistry is important for the compatibility of lead, and maybe the incompatibility of lead when you recrystallize monazite, but the compatibility of rare earths with respect to that mineral. So this is all great, but this is just one complex in Australia. But Australia actually has a lot of complexes, both carbonatite and just rare earth rich deposits in general. And so what Carl and Timur and I did was we started plotting this. So this is again Yangibana, but we started plotting this up for other examples as well. And sure enough, in No One's Bore, in the center of Australia, which is really the guts of like the very yeah, really the guts of a carbonatite. It had some age issues as well, but when you plot everything up with its isotope values, you can say, okay, there is a singular event and it evolves along this line. And then we can, again, using isotopes, go back and be like, okay, well, this is when it all happened. So using the isotopes, we can kind of find out what fertilized the mantle to allow for rare earth enrichment later on, and then what's happened after that. And so when you take all those deposits in Australia and you start looking at that, you can do the exact same thing. You can see, oh, there's these big orogenic events. The Glenberg orogeny, for example, is a subduction. It was some continental subduction going on in the Western Australia craton. And you've got all these big collisions and separations and stuff like that that are fertilizing the mantle. You get the formation of the deposit, and then afterwards you can record anywhere from a couple hundred to over 1 billion years in time. So really all up, rare earth deposits and carbonatites in particular are really important, because you can look to see the cause and effect and what happens after you've then placed this essentially this magmatic intrusive. And the same ore minerals that are really good and make them valuable critical resources are really good tools for kind of looking before and after what's happened. So we did this with carbonatites, paralkaline deposits, skarns, IOCGs, and I really don't see any reason, like with some of the iron oxide apatite deposits kicking around Europe, why we couldn't do something like that. Thank you.
Thank you, Paul. That's a fascinating look back into these large tectonic events. Do we have any questions for Paul?
Yeah, we have Murray there. He has his hand up. Murray, do you want to ask a question?
Sure, thanks, Paul. Nice talk. So one of the issues, it was interesting you brought up Bayan-Obo, because the dating at Bayan-Obo, as you probably know, is all over the place as well. Have you done the same sort of analysis to look at all the different dates that have been produced from Bayan-Obo?
Yeah, that's so when you take all those dates, and this was done the same time we were publishing our work, if you take them and you shoot them back through time, they all coalesce at one age, and that's that 1300-ish age. So it's not, so really a lot of the 30 years of dates, and they are everything from 1300 down to like 400 million years. You can pretty much do the same thing. You just need to have some confidence in kind of your really your bulk rock isotope values, and you just start plotting stuff like that. And we've been able to do it in a few different places, so I don't really see why it can't be used elsewhere for issues going forward.
Great, and yeah, it looks like we've got no more questions coming through there. So unless Betty, you've got something for Paul, thanks very much. And Betty, I think you might be on mute there.
Just quickly, Paul, I was interested to say about the techniques you were doing in situ in the field. Could you just briefly say something about those again?
Well, yeah, sorry, that's my mistake. They weren't in situ in the field, they were just in situ in the rock. Like, I had to take the rocks back, but I didn't have to separate them or clean them or do anything else. I get all that context in a thin section or a puck. No, that would be great if we could take a multi-collector into the field.
Oh, you had something really.
No, I didn't clarify. That's my fault. But that's.
No, that was brilliant. Thanks very much.
There's one more question there just before we go there. Betty just came through from Gary O'Sullivan. Hi, Paul. Cool talk. Are there any trace element patterns common to the various monazite age populations?
There. Like anything that separates them within the deposit, or anything like, I guess if you could elaborate a bit more.
Sure, your email is there if he needs to get in touch.
Yeah, I was to say they've all got very similar rare earth patterns. There's a bit of change that happens with lanthanum and neodymium, but that's something a little outside of this talk. Not. So yeah, that's something we could talk about further. It gets complicated very quickly.
I'm sure it does. Okay. Grant.
But I'll hit Gary up with an email here real quick. Thank you. Sorry.
Thanks, Paul. And now we're moving to our final talk of the day, last but not least, and a very interesting title. It looks like it should be something for the barbecue, I think. We have lightly salted tetrapods. Do Pennsylvanian coal swamp assemblages preserve a brackish ecosystem? And we have Adon Ogo and his team going to present on this interesting topic. Thank you.
So is my screen up there now?
Yeah, it is, yeah.
Okay, grand. So tetrapods are the group of animals that include all terrestrial vertebrates. So it includes reptiles, mammals, birds, and amphibians. And they first appear in the Devonian with trace fossils preceding actual body fossils. And all these Devonian tetrapods were really restricted to aquatic environments. Not a lot of them could actually get up and go about on land. And these were decimated by the double mass extinctions in the Devonian. Now, going from the Devonian into the Mississippian, we get this transition from a hothouse world into an icehouse world. And our fossil record of tetrapods in the Mississippian is kind of poor. It is getting better. However, there does appear to be this kind of diversification among tetrapods during the Mississippian. And it's during this period that we get the first truly kind of terrestrial forms that could actually get up and go about on land. Now, the Pennsylvanian, on the other hand, has a fantastic rock record for tetrapods. And we have a huge amount of assemblages that preserve tetrapod communities from various different ecosystems. The most famous of these are the ones that are preserved within coal seams or in association with coal seams, which are often termed the coal swamp assemblages. And that's what we will be looking at here. So these preserve larger statin quality tetrapods. And here's some images. They're not fantastic, of some of the tetrapods fossils from one of these coal swamp assemblages. Now, there's a long history of work done on these Pennsylvanian coal swamp assemblages. And reconstructions typically look something like this, where you have this inland bog, you've got a few insects flying about, and there's some tetrapods lounging around on the banks of a bog or a swamp. A lot of these coal swamp assemblages have traditionally been considered to preserve freshwater communities, and this is based on two facts. The first is that you're getting a high abundance of plant material, and true enough, plants don't grow in the sea, they're only going to grow in land and freshwater environments. But the other indication that these are freshwater environments is that people have often traditionally associated early tetrapods with modern amphibians. And modern amphibians only live in freshwater environments, with the exception of one frog. However, it is apparent that modern amphibians and early tetrapods, although they do share some characteristics, don't share a huge amount in similarity when it comes to physiology. So we have to think about them as a kind of separate animal group. Now, although you often see these reconstructions of these coal swamp assemblages associated with the Pennsylvanian, the Upper Carboniferous, and despite the fact that we have a huge amount of coal seams preserved within the Pennsylvanian strata of northeastern America, there's actually only 4 coal seams that preserve these coal swamp assemblages. Those are the assemblages at Linton, Ohio, Jarrow in Ireland, Newsham in England, and near Shannon in the Czech Republic. And based on the morphology of these coal seams, they can be grouped into 2 different categories. At Linton, Jarrow, and Newsham, the coal seams all thicken locally into a channel, which is between 100 to 300 meters wide and usually around 1 kilometer long. And these are called the channel coals. While as at Nirani, Nirshani, there doesn't seem to be any channels forming. So the coal here has been interpreted as being deposited in a lacustrine environment. Now, the coal seam Nirshani is deposited in an intermontane basin. So it actually does likely preserve a freshwater assemblage. However, all 3 channel coals are deposited in paralic basins. and the interpretation of them as preserving freshwater assemblages doesn't really hold water. So the traditional model for how these channel coals were deposited is that they were deposited within abandoned channels or oxbow lakes, and that the tetrapods that you get preserved in them represent a tetrapod community that were living within these abandoned channels and oxbow lakes, whereas coal sourced within the channel is autochthonous and formed from plants that were also growing within these lakes. However, there is some slight issues with this traditional model. The first is that while I don't know if that's blocking, while the coal seam at Linton and Jarrow do appear to form channels, the coal seam at Newsham actually tends to forms a dome. So it actually domes into the strata overlying it. So it doesn't really form a channel and more of a dome. The second problem with the model is that a review of coal from within the Jarrow channel in the 1980s found that it was actually made up by mostly washed in material. So it's not formed from plants that are growing within a lake. It looks like this is an area where organic matter is collecting. And it's likely that the tetrapods and other fossils that are preserved within this assemblage have also been washed in. And the final issue with this is that the tetrapod fossils that are found within the coal seams in the Linton assemblage, they are found within the channel. But at Newsham and at Jarrow, they're actually found in the bituminous shales that overlie the channel. So although they are relegated to the channel, they aren't actually found within the channel. Now, in the Pennsylvanian, coal forms during a late transition or a late transgressive system tract. And in often cases, a lot of these Pennsylvanian coal seams are capped by a marine deposit, which marks maximum marine flooding. So this could be a marine limestone or a shale with marine taxa in it. And this marine cap coincides with the horizons at Jarrow and Newsham. Does that mean that they record a marine ecosystem? Not on its own, because, well, it's not indicative just on its own that it's going to be recording a marine influenced environment. And to look at what sort of paleosalinity this assemblage was deposited in or represents, we really need to look at the fossils. And to do that, I'm going to focus mostly on the Jarrow assemblage because that's the assemblage I work on. And with fossils, the best indicators of paleosalinity are going to be the invertebrates. Because for want of a better word, invertebrates tend to have a simpler physiology. It's more understandable what environments they can tolerate and what environments they can't tolerate. At Jarrow, we have two invertebrates. The first is this horseshoe crab here called Bellinurus. And this was reported from within the Jarrow assemblage when the fossils were first found in the mid-1800s. However, I've never actually seen a specimen of this horseshoe crab that looks like it's from within the Jarrow assemblage. It could be from the overlying shales or the underlying shales. Modern horseshoe crabs all live in marine environments. However, during the Mississippian and the Pennsylvanian, there was this group of horseshoe crabs that lived in freshwater environments, and this includes Bellinurus. So if Bellinurus is actually part of the Jarrow assemblage, we have an indicator that it's kind of more freshwater environment. The other invertebrate that we find in the Jarrow assemblage are these microconchid worms, which leave behind these spherical tubes. These were traditionally called Sporobus, but they've been reclassified recently. And you definitely find these within the Jarrow assemblage. So on the bottom right here, I have a CT slice of the vertebrae of one of these tetrapods from the Jarrow assemblage. And you can see this spiraling microconchid worm tube beside it. And there's a 3D model of the worm tube just above there. And microconchid worms are found in marine deposits. On the pictures on the bottom here, I have a picture in hand sample at A and B of a limestone. And you can see it's full of these worm tubes. And then from C to F, we have a thin section of this limestone, and it's full of brachiopods and crinoids. So this is irrefutably a marine limestone. And it's from roughly the same age as the Jarrow as well. So the idea with microconchid worm tubes is that you get them preserved in brackish environments where they have been washed in from the marine realm. So they don't necessarily suggest a marine environment, but they do suggest a brackish environment where you're getting marine wash in. Right. So although the channel coal assemblages are famous for the tetrapods that they preserve, they actually have a higher diversity in species of fish material and even more specimens of fish material. So here mapped out are the different major fish genre found within the three channel coals. So on the group on the here on the left, we have the chondrichthyans, and these are the cartilaginous fish such as sharks. And then we have the sarcopterygians, which are the lobe-finned fish, and this include the likes of the dipnoa, which are lungfish, and the rhizodonts. I've left out actinopterygians or ray-finned fish because their systematics for Carboniferous taxa is kind of all over the place, and it's very hard to actually identify which taxa they are. And one thing to note is that the assemblages roughly have similar kind of fish fauna. What's really interesting is that in the assemblages, you're getting either Xenocanthus, Triotis, or Orthocanthus down here, which are these sharks. And these sharks belong to a group of sharks called the Xenocanthiforms. And these have been traditionally considered to be strictly freshwater sharks living in lakes or rivers. However, you find their teeth in huge abundance and even fossils of them in marine deposits. So it's likely that they are actually euryhaline, and that they can live in both freshwater environments and in marine environments. And we saw in one of the posters today that you're actually getting these xenocanthiform teeth in one of the limestones down at Brendan's Well. Now, one thing to note about the fish fauna within the channel coals is that you find pretty much all of them also preserved within brackish or marine rocks as well. So on the right here is a diagram showing the ranges of some of the typical Mississippian and Pennsylvanian fish. And where they're marked with a black dot is where they are found in a fossil assemblage that was deposited in a marine to brackish environment. And you see that fish such as Saganotis, the lungfish Saganotis, and all the rhizodonts that you find within the channel coals are also found preserved in these marine and brackish environments. Another thing to note about the fish fauna is that if they did represent a freshwater community, you'd expect them to be endemic. You'd expect to find species and genre that only occur in one or two localities. Whereas the fish fauna for the Jarrow coals is quite cosmopolitan and is found in a variety of different environments, or is found in different variety of different fossil sites throughout the Carboniferous. So on the right here, these are fossil sites in the Maritimes Basin in eastern Canada. Another thing to note is that the fish fauna is made up of large predatory fish, and it's very unlikely that if you have a cut-off oxbow lake, that you're getting a huge diversity of large predatory fish within it, because an oxbow lake of 1 kilometer long and 200 to 300 meters wide isn't going to really preserve that diverse community. There won't be enough nutrients to sustain it. What can the tetrapod fauna tell us about it as well? So at Jarrow, the tetrapod fauna is dominated by 4 different groups. These are the limbless aestopods, the newt-like nectrodines, and the long-bodied and short-limbed coleostids and embolomirs. Now, it's been considered that you can actually break up tetrapod faunas based on what sort of environment they lived by looking at what groups are dominant. And it's been suggested that brackish tetrapod faunas are dominated by coleostids, embolomirs, and a group of nectrodines called the diplocolids, and that's what we see at sites like Jarrow. Whereas freshwater tetrapod faunas are dominated by disaurophid temnospondylus and microbrachids. Now, there was a single specimen of microbrachid identified from the Jarrow, but this was imaged using CT scanning, and it turns out it actually is a diplocolid nectrodine. So the Jarrow tetrapod fauna looks like it's more towards the brackish side of things than the freshwater side of things. And here's the range of major tetrapod groups throughout the 4 assemblages. So Jarrow, Newsham, and Linton are the channel coals, whereas Nirshani is this lake coal deposit. And the first set of pie charts on the right record the number of species per each major group. Now, this is a bad indicator of which tetrapod groups were dominant, because you're going to have washed in elements. You're going to have some tetrapods that have been washed in. So some of these species are only known from a single species, whereas other tetrapods are known from several hundred species. So on the right, the pie charts on the very right record the number of specimens per each major group. Now, I actually couldn't get any data for the Nirshani assemblage, so that's actually, I couldn't really plot that out. However, based on the literature, that is dominated by microbrachids and dicerophid temenous spondylus, which again are these kind of more freshwater tetrapods. Whereas the Linton, Newsham, and Jarrow assemblage are all dominated in some form or another by embolomirs, coleostids, and diplocollid nectridines, which are these more kind of brackish taxa. And here is a cladogram of early tetrapods with the 4 major groups seen at Jarrow highlighted here. And where the branches are black, this is where you find these tetrapods preserved in freshwater environments. Whereas where the branches are coloured in green, these tetrapods have actually been found in brackish deposits. So you can see that not only are the Jarrow, or the main groups that are seen at Jarrow, being preserved in brackish environments, it's very likely that a lot of early tetrapods were preserved in brackish environments. And this suggests that a lot of early tetrapods were euryhaline, so they could tolerate a variety of salinities from freshwater down to marine. And this isn't surprising when you consider the fact that they originate from a group of fish called the rhizodonts, which also show this adaptation. So it's likely that a lot of tetrapods retained this euryhaline adaptation. Now, in the Pennsylvanian, the coal swamp assemblages were all deposited in tropical areas. And glacial-induced sea level change caused the flooding of lowland areas, which created these shallow seas. Now, many of these shallow inland seas may have been slightly brackish, similar to what we see in the Baltic today. And it's likely that this euryhaline adaptation may have been a huge benefit to these tetrapods that were living along the coastlines of these environments. It's also worth noting that if the Jarrow assemblage is brackish and represents euryhaline taxa, they probably had a more kind of cosmopolitan distribution, and therefore the coal swamp assemblages aren't a just snapshot of a isolated lake community, but maybe more representative of the kind of typical tetrapod fauna along the coastline of the tropics of the Pennsylvanian. So instead of thinking of these coal swamp assemblages as being these inland bogs or swamps with tetrapods lounging about them, a better modern analogy might be something like the mangrove forests in tropical areas like Florida and Brazil, where you get a mixture of kind of freshwater and marine taxa living in estuarine environments. And that's everything from me. Thanks very much for listening, and I'm more than happy if you have any questions.
Thank you very much, Aidan. It's very interesting to see the amount of data that is on the tetrapods that you've captured. Have we got any questions, Luke?
No questions coming through here on Teams or YouTube.
So I suppose one question that comes to mind from my past study days is the lingular brachiopods, we were told, went right through the geological column and represented brackish water. Do you find those associated with the tetrapods?
No, so the main problem with a lot of these tetrapod assemblages is that even though they preserve a huge diversity of vertebrate material, invertebrate material is actually pretty rare. So you do get lingula bands above and below coal seams, but because you don't actually get them in the same horizon as the tetrapods, you can't really use that to infer whether they're brackish or freshwater. They are useful for indicating that it is a peralic basin, though.
Yeah. Is it difficult to decide if the remains have been washed in, or that they were kind of living in that environment?
So I think it's likely that they are washed in, because you're getting this, if you're getting, with the gyro, if you're getting the kind of plant material washed in, it's likely that the tetrapod and the fish material might be getting washed in as well. And the whole problem with the idea of this kind of diverse tetrapod community living in a lake is that all these tetrapods are still pretty much aquatic. Not a huge amount of them are going to be going about on land. So they need to access this lake via water. And if it's a cut-off lake, you wouldn't expect such a diversity of different animals living together, especially in such a kind of small lake.
Yeah. Okay, so if we have no other questions, Luke, nothing coming in yet?
No, nothing. Well, I might ask maybe a basic question. I think we have a small bit of time. You mentioned at the very start here, my knowledge on tetrapods is somewhat poor. You mentioned diversification occurred in the Mississippi, and why exactly was that?
Oh, not 100% sure. So I guess in like post-Devonian mass extinctions, ecosystems might have opened up. Also, in the Devonian, you're starting to get other animals colonizing terrestrial environments. And this is kind of enriching the ecosystems in maybe freshwater and estuarine environments. So maybe it's a reflection of that. It's not kind of fully understood what's actually driving the diversification, but you also see a diversification in fish after the Mississippi. And so it's the kind of typical post-mass extinction diversification of animals.
On the ball. Yeah, I don't think there's anything else here coming through.
Well, thank you, Aidan. And
all right, thanks very much.
I think that completes our session, our fourth session. And I want to thank the four speakers and their colleagues who they work with, and for the questions that came in, and thanks to all our people who've tuned in. And I want to just give a particular thank you to Brendan, who's organised all of this for us today, and the keynote last night. And I'm going to pass over to him now to say a few closing remarks and for a special presentation.
Okay, thank you very much, Betty, for chairing that session, and to all the speakers and everyone watching. The one thing that we need to do before we finish up is the IGA have very kindly sponsored prizes for the best student talks and posters, and we have had Michelle Castieux, Laura Smith-Egan, Angela Colcase, and Mike Sims judging the talks and posters. So now I'll pass you over to Michelle from the IGA just to announce the winners.
Okay, thank you, Brandon. So I'm not sure do I need to spotlight, or is that okay like this? Yeah, can you see me? That's fine. Yeah. Okay, so just want to say hello to everyone. So like Brandon said, my name is Michelle Castieux. I'm the current president of the IGA. And I won't speak for a long time. I just want to say the IGA is open to all who have an interest in geology, whether it's professional geologists or beginners. We do lectures and field trips. Obviously, at the moment, it's only Zoom lectures. But hopefully we'll get back to being on site very soon. And keep an eye on our website, geology.ie, or our Facebook page for events. So I just want to sincerely thank the judges for their time. But most importantly, I must say that the posters and talks were of a very high standard, and it was very difficult to come to a decision. But we have to make a decision, and we could see that the participants put a lot of hard work into this. I want to congratulate them all for their work, their dedication, and especially given the difficult circumstances that we've had since the pandemic began. I am honored to award the best talks and best poster prizes on behalf of the IGA, and I'm going to give you the results now. So for the posters, firstly, we also have some commendations because like that there was so many good talks and presentations. So we have a couple of commendations for the flash talks. The first one is Emer McKenna with the geology of the Carlingford Igneous Complex, and the second one, Sean Houghton with the catchment characterization hydrology of the Barna River in County Galway. So that was for the flash talks. Then we have a couple of commendations for the posters. So we had Orla Tyrrell with the geology of the Glentrasna area, and Larissa Macedo Cruz de Oliveira with use of 3D photogrammetry for advancing seabed mapping techniques for deep water habitat classification in submarine canyons. So well done. Now there are prizes for the posters. We actually decided on 2 first places. Okay, so there's no runner-up. We have joint first places. So the first one is Amy Bename, and I have to apologize for my pronunciation of any name, so I apologize for that. So Amy Bename with rainfall as a trigger for volcanic activity, analysis of historic eruption records. And the second one, Avery Fenton, new Mississippian fish, microvertebrates assemblages spanning a deepening marine environment from St. Brendan's Wells, County Clare. So that's for the posters. Now for the talks, so we have one commendation for the student talk. So David O'Leary with the potential of self-organizing maps clustering to characterize a harvested peatland using airborne radiometric data and a digital elevation model. And the comment from the judges was great talk, including a diverse range of methods and evidence, excellent diagrams, and very well delivered. So the 2 joint first place prizes as well, so no runner-up for the talks. So would be the first one, Benjamin Coven, emplacement and internal deformation of submarine landslides constrained by resistivity image log interpretation. So the comment was very well delivered and explained talk, diagrams and data excellently presented and discussed in a knowledgeable manner. And the other first prize, Adrienne Marie Sux, replacement of calcite by rare earth carbonates, the formation of bastonacite, some incredibly interesting, so the comment was some incredibly interesting images, very engaging speaker with experimental results presented accessibly. So well done to all of you. So the 4 prize winners will receive a €50 check from the IGA, and I think Brendan will be in touch with them for postal address and so on. So just finally, I would like to thank Mary Immaculate College for hosting this conference, but especially Brandon for an excellent organization. Okay, so I mean, I'm sure it was quite complicated given the situation. So well done. It's been a great IGRM. So thank you very much.
Thank you very much, Michelle. Let's see if I can stop spotlighting you there. Okay, before we all leave, there's one other thing that I do want to note. I didn't think that we should go through the conference without acknowledging someone who isn't here anymore, who has been a big part of that, and that is Matthew Parks, who sadly passed away earlier this year. Matthew was always a big part of the IGRM over the past few years, and just a comment that someone made to me earlier today, more and more it becomes apparent how much Matthew was involved in so many diverse geology projects around Ireland, and what a huge contribution he made to the geological community here. His passing is a huge loss to so many of us. I think a lot of us would agree with those sentiments, so I just wanted to just take a moment to mark that. So that is the end of the conference for this year. I'd like to again thank the IGA for sponsoring the prizes. I would like to thank Aoife, who organised last year, for the advice and support in getting the conference going this year. And of course, I'd like to thank our speakers, everyone who chaired sessions for us, and everyone who attended throughout, whether asking questions or just watching and enjoying. It was great to have so many people engaging, and I'm really glad that we were able to have an IGRM at all this year, despite the circumstances that we're in. Next year, the IGRM will be in Belfast, assuming that the health crisis has by then, and that we are able to have a meeting in person. So fingers crossed for that. If it does go back to an online event, don't count on me to do it again. Hopefully I will see you all in Limerick next time we'd host it, which by my count would be 2027, so I can have a break of a couple of years until then. So thank you very much to everyone. I hope you enjoyed it, and we'll see you next year in Belfast. Thank you.
Thank you. Bye.
Thank you.